remind us?
Yeah, I remember this morning. Class, my college. Small Division II school near Johnson City, Holocaust one.
Everything okay?
Well, I've got quite a few friends of mine who are outside facing an entire but that's what we
Yeah.
Jesse, report.
Huh?
Report.
Yeah. Well, do we want to go ahead and start?
Oh, I see. So as long as we have time, we're good. Okay.
Good afternoon, everyone. Thank you for coming. I know everybody has a busy schedule, but this is an important part of the work that we do. We are here to listen to Glenn Vicary, who is a master's degree student at the University of Louisville and who's now going to basically present his work for his Ph.D. Dr. Barber just arrived. That's good. So a little bit about Glenn. Many of you know him, but just a reminder, Glenn obtained his Bachelor in Arts at Tusculum College in Tennessee and then came to Louisville and joined University of Louisville Pharmacology Toxicology program and obtained his master's in 2013 and has been working in his PhD ever since in my laboratory. His area of interest has been in the area of lung fibrosis, so he's going to talk to you about lung fibrosis. And for that, you have to understand a number of concepts and master a number of in vitro and in vivo projects. He's going to show you some data related to animals. And along the way, at some point, Glenn started worrying about how does he apply this? How do we move whatever I've learned in the lab to understanding how people behave and how do we deal with their clinical problems? And he decided to take the Entrepreneurship Academy. And he's done a couple of internships with Applied Pharmaceuticals and Revon. And I've asked him to just tell you one slide about that at the end so you have a sense of how he's already pursuing his future, how he's moving what he's learned in the laboratory to perhaps in the management of clinical trials and so forth. So I leave you, Dr. Glenn Vicary, hopefully.
Hopefully, yeah. So, thank you everybody for coming today. As Dr. Gracious introduction, but I'll delve right into it about my dissertation work, which is the role of nicotine, alpha-7 nicotinic acetylcholine receptor in extracellular matrix remodeling and pulmonary fibrosis. I'll just give you a brief background on my research and then walk you through the 4 research aims of my dissertation work. Initially, some conclusions and future directions of my research. So I want to start off with just a CT scan of a healthy individual on the left and a patient with severe pulmonary fibrosis on the right. And as you can see, there's drastic differences. And that's because on the right, what you're seeing is severe obliteration of the lung structure. Marked, a characteristic of pulmonary fibrosis is this honeycombing that you can see in several areas of this patient's lung. Now, what happens with pulmonary fibrosis is as the fibrosis progresses, it obliterates all lung structure and the patient experiences loss of lung function, and most of these patients end up dying due to respiratory failure. But there are many forms of pulmonary fibrosis, but my research focuses on the idiopathic form. And the hopes is that by uncovering mechanisms in the idiopathic pulmonary fibrosis, you can translate to treat or better understanding of the other different forms of pulmonary fibrosis. Idiopathic pulmonary fibrosis affects over 100,000 Americans and is characterized by, as you saw on the last slide, fibroproliferation and excessive production disorder deposition of the extracellular matrix. It's diagnosed in older patients, with over 85% of the patients being older than 50 years old. Additionally, it's often detected late, which leads for a bad prognosis with an average survival of 3 years post-diagnosis. And additionally, it makes it difficult to study this disease because it is an unknown cause. As I stated earlier, there's different forms of pulmonary fibrosis which are associated with these exposures that you see on the right, including antibiotics, radiation, genetics, which about 4% of all pulmonary fibrosis is associated with genetics, acid reflux, and certain environmental exposures. But by above and beyond the rest of these, cigarette smoke exposure is the most highly associated cause of pulmonary fibrosis. Over 60% of idiopathic pulmonary fibrosis patients are either current or former smokers. And studying this scientifically is made very difficult because it's extremely complex. It contains over 7,000 chemicals, which makes it difficult to tease out what each individual compound does in the body. Some of the chemicals that cigarettes, I apologize for the flashing on the screen, some of the chemicals include hexamine, cadmium, and it's not only complex, but it's also a huge public health concern because 1 in Americans still smoke. And it costs the economy a large amount of money. If we could get 10% of the people who smoke to quit smoking, it would save the U.S. economy and healthcare expenditures of over $63 billion the next year. Now, as I said earlier, it's complex, which makes it very difficult to study scientifically, which is why, for my research, I focused on nicotine, which is the main alkaloid of cigarette smoke or tobacco exposure and is the main addictive component. And this has to do due to its high bioavailability, which means it distributes all over the body very quickly and very fast. It's been shown that nicotine can cross the blood-brain barrier and reach the brain within 7 seconds of inhalation. Not only that, when you breathe in cigarette smoke, it acts as a deposition center in the lung, showing up to 4 times higher concentrations of nicotine than what's seen the rest of the way systemically. And its effect is it is a stimulant at low dose, which is why you have the addictive effect, but it can be poisonous at higher doses. And recently, this has kind of resurged in the entrance of science because of the rapid rise in the use of these e-cigarettes, primarily among young adults, and where nicotine is one of the main components of e-cigarette solution. Now, nicotine interacts all over the body by its class of receptor called the nicotinic acetylcholine receptor, which is a multimeric acetylcholine triggered cation channel made up in a pentamer form, so 5 subunits. And they've been seen on several cells over the body, including the immune system, epithelial cells, and fibroblasts. They can be made up in a heteromeric form, which means multiple different subunits, such as in the alpha-3 or the alpha-5 forms of this receptor. But the alpha-7 receptor is the most abundant homomeric form. So it's 5 of the same subunit. And this is what my research is focused on, is the role of this receptor. Now, because it can distribute all over the body, and these receptors have been seen all over the body, there's previous research has shown nicotine has a wide variety of effects on cellular processes, including increased cellular proliferation and tumor vascularity dealing with cancer. It also can upregulate its own receptor. And lastly, because it can cross the blood-brain barrier, it can also cross the placenta. It's been shown to cause prenatal lung tissue remodeling in infants. So now that we covered the background, we'll dive into the first aim, which is the effects of nicotine on lung fibroblasts and the nicotinic receptors responsible. And the reason I'm focusing on lung fibroblasts is because they are the primary resident cell in the lung, which control extracellular matrix deposition and remodeling. And so this led to the first hypothesis of nicotine interacting through the alpha-7 nicotinic receptor to induce cellular signaling to induce extracellular matrix proteins and also fibroblast proliferation. And the first extracellular matrix protein we looked at was fibronectin, which the Roman lab has had a lot of history researching and looking at the role of this protein in several diseases. And fibronectin is a glycoprotein that has roles in cellular adhesion, growth, and migration, and it's critical in initial wound healing. In this cartoon on the right, fibronectin is represented by these little green strands that you see, with this being the cellular membrane below and the extracellular matrix above. And the reason it's critical in initial wound healing is it helps to potentiate cell adhesion and cell interaction with the extracellular matrix during initial wound healing. And when we began to look at what would nicotine expression or nicotine exposure do to fibronectin expression, previously our lab had shown that nicotine has the capacity to induce ERK, which is a protein in the MAPK kinase pathway, which affects several cellular processes, both the phosphorylated form, which is the active form of ERK, and total ERK. And also we showed that nicotine induced significant amount of fibronectin expression, but when using a MEK1 ERK inhibitor, so blocking this pathway, we were able to block this induction caused by nicotine, showing that nicotine is inducing fibronectin, but it's through the MEK1 ERK pathway. Additionally, as preliminary part of my research was looking at the nicotinic receptor involved in fibronectin expression. And what I found was using wild-type fibroblasts and exposing them to nicotine for 24 hours, we found significant upregulation of fibronectin protein, shown here by Western blot with the quantification below, but we also used fibroblasts from mice lacking the alpha-7 receptor, so alpha-7 nicotinic receptor knockout mice. And what we found was fibroblasts from these mice, nicotine did not induce fibronectin, showing that nicotine is interacting with the alpha-7 receptor to induce fibronectin. Now, as I stated earlier, it's a protein involved in initial wound healing. So the next part of the research was looking at collagen type 1, which is a more permanent protein, which it's more structural and more lasts longer than fibronectin in the extracellular matrix. And so we looked at collagen type 1, which is the most abundant form. It makes up over 90% of collagen in the body. Similar to fibronectin, it does have increased deposition and injury, but it is a little bit more of a permanent protein and can affect things such as tensile strength of the extracellular matrix. So when we exposed fibroblasts to nicotine, what we found was similarly to fibronectin, was nicotine induced a dose-dependent response of collagen type 1 expression by mRNA. Additionally, we looked at protein expression after 24 hours, found increased protein expression from fibroblasts, showing that nicotine is inducing collagen type 1, both mRNA and protein. Additionally, using the same alpha-7 knockout fibroblasts from earlier, we showed that nicotine was unable to induce collagen type 1 mRNA expression and protein in these with exposure to nicotine. Again, showing that nicotine is inducing collagen type 1 similarly to fibronectin through the alpha-7 nicotinic receptor. The next focus was to look on proliferation. How does nicotine affect fibroblast proliferation? And it is a complicated slide, but bear with me, I'll walk you through with it. This first white bar seen here, here, and here is control untreated cells. The second bar that you see here, and here, and here is cells, fibroblasts treated with nicotine. And as you can see, at both 48 and 72 hours, there was a significant upregulation of proliferation in these cells with exposure to nicotine. We investigated the role of the alpha-7 nicotinic receptor in mediating this effect, and shown here on the black bar is cells treated with alpha-7 siRNA to knock down the receptor treated with nicotine. And at 48 hours and 72 hours, we were either able to block the effect or significantly decrease the amount of proliferation induced by nicotine exposure. Additionally, we looked at the cellular processes or pathways involved and used the same MEK1 ERK inhibitor from earlier, PD5059, and marked on this schematic as MI. And what we found is, again, nicotine induced significant proliferation at 48 and 72 hours, but presence of an inhibitor blocked the proliferative effects of nicotine. And so this showed us that nicotine is indeed inducing proliferation and extracellular matrix production through the alpha-7 nicotinic receptor in the MEK1 ERK pathway. Now, everything I've shown you so far is in vitro. So, what the question is the same thing seen in vivo. So, to be able to look at this, we used a chronic nicotine exposure model, which was actually developed by Dr. Rao and our fabulous department several years ago, which involves taking mice and treating them with nicotine at 100 micrograms per ml in their drinking water for 90 days. And this achieves plasma levels seen or equivalent to what's seen in humans and heavy smokers. And after exposing these mice for 90 days of nicotine, indeed what we found was similar to what we saw in vitro, which was that in wild-type mice treated with nicotine, we found significantly upregulated fibronectin mRNA when compared to untreated control mice. Additionally, using alpha-7 deficient, nicotine receptor deficient mice, we found that this effect was absent in these mice, showing that nicotine indeed was inducing fibronectin expression through the alpha-7 nicotinic receptor. Now, here on the right is immunohistochemistry staining for luciferase. So we have mice with a fibronectin or with a luciferase gene connected to the fibronectin promoter. So, and we use this model to investigate what cells were affected by nicotine exposure and what cells were having increased fibronectin, and was it deposited the same way. So luciferase staining is brown. So as you can see, you see increased amount of staining in the right two cells, which are the nicotine-treated mice. Not only that, you see positive staining all over the lung, showing that there are multiple cell types that are inducing fibronectin, and it is being laid over normal lung structure. Next, we began to look at collagen, and what we found was that, again, nicotine did induce significant increase in collagen type 1 whole lung mRNA expression, and this induction was absent in the alpha-7 knockout mice. Additionally, looking at protein expression, we found increased levels of phosphorylated ERK, phosphorylated SMAD, and phosphorylated collagen, shown here by quantification. And the reason we looked at phospho-ERK based on earlier, seeing if this was mediating the effect, and also looked at SMAD3, which is a major protein in the TGF-beta pathway, which is a profibrotic pathway, but it's also been shown to be involved in pro-inflammatory responses. And lastly, we found significant upregulation of collagen type 1 protein. Next, we looked at tissue staining by immunohistochemistry, or not immunohistochemistry, but histology. And we used Mason's trichrome staining, which in Mason's trichrome staining, collagen stains as a bright blue. And what we found was increased collagen staining, shown here, but highlighted by this red arrow, increased blue staining, primarily around the major airways and blood vessels of the lung, and this was significantly increased when compared to PBS or control-treated mice. Additionally, we used alpha-7 knockout mice and found no differences in collagen staining between wild-type and nicotine-treated alpha-7 knockout mice, showing that its ability to induce this collagen type 1 and fibronectin was specific to the alpha-7 nicotinic receptor. Next, now that I've shown you that nicotine is inducing an altered extracellular matrix, we're going to look at how could this alter protein composition? Could it affect other cellular processes or other cells? And so we're going to look at whether it's possible implications for inflammation. And to do that, we use an isolated fibroblast matrix model, which involves taking fibroblasts and culturing them for 5 days in a culture dish, and then lysing them via osmotic lysis, which removes all cellular material while still leaving the extracellular matrix on the bottom of the culture dish. We then took human monocytes, U937 cells, and cultured them on top of this matrix for 24 hours and looked at interleukin-1 beta, a pro-inflammatory cytokine, for the expression. And indeed, what we found was that treating the fibroblasts on this step for 5 days with nicotine, their matrix caused a significant increase in interleukin-1 beta response in the monocytes, showing that nicotine was able to induce a pro-inflammatory response independent of direct mechanism, just by a change in the extracellular matrix alone. The next step was to look at, is this effect mediated by the alpha-7 nicotinic receptor? And indeed, what we found was, again, nicotine exposure on the fibroblasts induced significant interleukin-1 beta response, but using either an alpha-7 antagonist, so blocking the alpha-7 receptor, or using fibroblasts lacking the alpha-7 nicotinic receptor, there was no induction in interleukin-1 beta, showing that this pro-inflammatory matrix induced by nicotine is controlled by the presence of the alpha-7 nicotinic receptor. The next step was to look at possible pathways involved. So we looked at the involvement of ERK signaling, but then also, more importantly, looked at the mechanism by which this modified matrix was inducing interleukin-1 beta response. And to do this, we looked at integrins, which is one of the main receptors which cells interact with the matrix. And what we did was looked at the alpha-2 beta-1 integrin, which is the main integrin by which cells interact with collagen in the extracellular matrix. And using this alpha-2 beta-1 inhibitory antibody, we were able to block the induction of interleukin-1 beta when compared to the nicotine-treated fibroblasts alone. Additionally, what we found was using the MEK1 ERK inhibitor used from earlier, we were able to achieve the same effect by blocking the increased interleukin-1 beta induced by treating fibroblasts with nicotine. So furthermore, we looked at whether this effect was valid in vivo. And indeed what we found was mice treated with nicotine had significantly more interleukin-1 beta expression in whole lung mRNA. And we also did immunohistochemistry staining for interleukin-1 beta with PBS-treated mice on the left and nicotine-treated mice on the right. And as you can see, there is increased interleukin-1 beta staining. In addition, you could probably spot more of these brown dots, which we've not quantified this, but we believe it's macrophages, and it is recruiting induced macrophages, which is involved in the initial inflammatory response. So that leads to the summary of the first aim of my research, where we show that nicotine acting through the alpha-7 nicotinic receptor and ERK signaling induces cellular proliferation and this altered extracellular matrix with increased fibronectin and collagen type 1, and that this matrix independently can cause a pro-inflammatory response. And this kind of got us to wonder and investigate what is the role of the transitional matrix or this altered matrix composition. And this figure was adapted by the wonderful Dr. Artiel here in the audience today. And we think that this altered matrix composition, so increased fibronectin and collagen type 1, may be a compensation mechanism for subtle challenges such as nicotine. And that in normal repair, this increased protein or altered protein composition would get resolved back to a healthy lung. But what happens if you're exposed to a secondary injury or an insult? Is this what causes fibrosis? And does it increase fibrosis progression or decrease lung function? What are the effects of this transitional matrix with the formation of pulmonary fibrosis? And that was the focus of my second aim, which is the effects of chronic nicotine exposure on injury-induced fibrosis. Now, one thing that I've shown you is I've shown you lungs treated with nicotine, and you're seeing increased collagen type 1 and increased fibronectin exchange, but the structure still looks the same. That's because to date, nicotine has not been shown to induce fibrosis independent of a secondary injury or secondary exposure. So what you're seeing is subtle alterations, but not complete change, or you're not seeing the formation of pulmonary fibrosis. And that creates a problem if we're trying to look at the role of nicotine in formation of fibrosis. So to be able to look at this, we had to use an inducible model of pulmonary fibrosis, which the most commonly used model in the scientific field is bleomycin, which is an anti-cancer drug that has a bad side effect, causes pulmonary fibrosis. And in using intratracheal instillation, for the first 2 weeks after instillation, mice experience a robust inflammatory response. Also, you get induction of profibrotic mediators such as TGF-beta. After the 2-week period, the normal lung structure is replaced by progressive fibrosis. And you can see here by the histology slides on the right panel is bleomycin-treated lung, and you can see the formation of fibrosis on the bottom part of this lung. I know it may be a little difficult to see in the back there. But there is a lot of benefit to this model. It does have several characteristics of human pulmonary fibrosis, and it does have heterogeneity, so differences in lung structure, where you might have this fibrosis area right next to normal lung architecture, which is what the same type of features you see in the clinical or human disease. So to be able to look at the role of nicotine in this fibrosis progression, we expose mice to nicotine for 90 days to form this transitional matrix, and then expose these mice to bleomycin and looked at the amount of fibrosis that formed in these mice. And as expected, what we found was nicotine did cause significantly more fibrosis in mice treated with bleomycin when compared to bleomycin alone, shown here by this is a bleomycin alone and nicotine and bleomycin treated bleomycin and nicotine treated mice. Top panel is control and nicotine to show you that there are no alterations in the lung structure with nicotine alone. Additionally, using alpha-7 knockout mice, we saw that this effect was absent. in alpha-7 knockout mice treated with nicotine and bleomycin when compared to wild type. But unexpectedly and confounding was that alpha-7 knockout mice treated with bleomycin alone had increased fibrosis when compared to wild type mice treated with bleomycin alone. And this suggests that the alpha-7 nicotine receptor may play several roles in the formation of bleomycin, but also at a steady state, showing that just the presence of alpha-7 may be driving, or absence of alpha-7 receptor might be driving increased fibrosis. So now that we've shown nicotine does drive increased fibrosis, but the role of alpha-7 nicotine receptor is unclear, we wanted to look at some other potential transitional matrix effects that could be different. And the one we began to look at was tissue stiffness. And the reason we did this is because there is new research suggesting the importance of matrix stiffness or the environment at which cells are being grown in. And this is because in vitro, cells are often grown in plastic and glass, which is way over here on the right at the 2 to 4 gigapascal range, which means very stiff and very hard. Whereas on the left, you're seeing lung tissue down below 50 kilopascals, often 1 to 10. And there's a lot of research that's shown lungs interact differently depending on the environment they're grown in. And this has been looked at clinically by Eric White out of the University of Michigan, who took lungs from normal patients and lungs from IPF patients and looked at the stiffnesses of those lung tissues to see if that's what affects the breathing mechanisms and the restrictive breathing seen in pulmonary fibrosis. And indeed what he saw was increased stiffness in IPF patients. Now, the important characteristic we wanted to look at, is this same effect valid with nicotine treatment alone? So nicotine inducing increased fibronectin and collagen, is that enough to cause significant difference in lung tissue stiffness? And we're still investigating it, but I'm going to show you some preliminary data. But the thing I want to point out is we're talking between 1 and 10 kilopascals. It's a very small difference. So we're not really sure if we're going to be able to see a difference with nicotine treatment alone. So to be able to look at this, we adapted a lung decellularization method, which involves taking lung tissue and removing all cellular material, which involves taking both the heart and lungs out as one together and then instilling different solutions, whether it's detergents, acids, or DNases, both through the heart and through the trachea, and different incubations. And what you end up with is this white cotton-looking thing here on the right, that is a mouse lung that is lacking all cellular material, but retaining its 3D extracellular matrix structure. And after doing some lung slicing, you see control lungs or normal lungs here on the left and bleomycin-treated lungs on the right. And you see these increased areas of bright white, which is increased protein concentration. And so we're not only seeing fibrosis, but you're seeing the heterogeneity of the fibrosis in these bleomycin-treated lungs, where you're seeing areas of normal tissue surrounded by areas of fibrotic tissue. So to verify that all cellular material had been removed, your top 3 cells are control-treated lungs, or control lungs, and the bottom 3 are lungs that have undergone decellularization. And shown here by H&E staining and trichrome, extracellular matrix proteins were still staining correctly, and additionally in the last 2 cells is fibronectin, which is staining as green. But in the top cell, you see these bright blue dots, which is DAPI, which stains cell's nucleus. And you can see a complete absence of blue staining in the bottom, suggesting that all cellular material had been removed while the extracellular matrix was still present. And the next step, in collaboration with Dr. Susi, we used atomic force microscopy, which is a very sensitive microscope which involves sticking a cantilever down on the sample and pushing down to determine how hard lung tissue or how hard this tissue stiffness is. And this allows us to look at generate what's called a force map, which each one of these squares represents a point at which the cantilever pushed down on the lung. And this allows us to look at both the overall lung stiffness changes, but also the heterogeneity of a lung, where you might get soft areas shown here by the black next to hard areas shown here in these white cells. In the preliminary data we've generated so far, we've only done bleomycin and nicotine treated lungs. But what we've found is that although there is an increased trend in the nicotine-treated bleomycin mice when compared to bleomycin alone, there is no significant difference due to the large heterogeneity of the readings. But further work, including control and nicotine-treated mice, are being conducted right now. So that brings me to the summary of AIM 2, where we show that nicotine does have a positive effect in the progression of pulmonary fibrosis induced by bleomycin, but the role of tissue stiffness is still being investigated. Now, one of the issues with using bleomycin is because it is a cytotoxic drug, mortality is a problem with these. You do, even in normal baseline experiments, a lot of people have a certain amount or percentage of mice die. And this led us to investigate the potential role of nicotine, the impact of nicotine exposure on survival in bleomycin-induced lung injury. And to do this, we instilled a higher dose of bleomycin than fibrosis to induce death in these mice that had been treated with nicotine. Now, based on the research so far, or these data I've showed you so far, with the increased amount of collagen, fibronectin, the increased inflammatory response we saw by the transitional matrix, we hypothesized that nicotine would increase the mortality or death rate of mice treated with bleomycin. And surprisingly, we saw the exact opposite effect. We saw mice treated with bleomycin alone are shown here as blue, and mice treated with nicotine and bleomycin is shown in this bright green. And as you can see, there's a significant protection against death with mice treated with nicotine. And they were dying at about day 10 to 12. We also used alpha-7 deficient mice and found that there was no difference in the alpha-7 knockout mice, suggesting that nicotine's ability to induce protection against bleomycin-induced death was through the alpha-7 nicotinic receptor. So as we've shown that it's blocking, but we wanted to investigate, and we've seen that it's through the alpha-7 nicotinic receptor, but we wanted to see maybe why they were dying. We weren't really sure what was causing the death. And as I showed you earlier, they were dying at days 10 to 12, which is still very early during the inflammatory phase before you should be getting fibrosis. So the first thing we did was just verify that these mice were not dying of fibrosis, and indeed that's what we saw. All 4 groups had the same amount of fibrosis, and there really wasn't much present. So we didn't think these mice were dying of fibrosis. The next step was to look at pro-inflammatory cytokines. Indeed, what we found was that there was a significant upregulation of interleukin-1 beta, shown here in the white bar, and TNF-alpha, which are pro-inflammatory cytokines. And we also looked at levels of TGF-beta, which is a pro-fibrotic mediator. And what we found was nicotine alone did induce significant levels of all 3 mediators, and that bleomycin induced a significant upregulation of TNF-alpha. But both of them together, nicotine and bleomycin, did induce significantly more interleukin-1 beta and TGF-beta. Interestingly, we looked at these cytokines and mediators in alpha-7 knockout mice, and what we found was that, especially interleukin-1 beta, there was no response whatsoever in the alpha-7 deficient mice, or TNF-alpha. There was some increased TGF-beta, but that could be due to nicotine interacting with other receptors. So based off of this, we didn't think these mice were they weren't dying due to protection against inflammatory response, because otherwise these would have been decreased. The next step we looked at was, well, are they dying due to reactive oxygen species, which is another mechanism by which bleomycin induces damage and death of cells. And in this, when the lung is exposed to a challenge, it often generates ROS, such as hydroxyl radicals or oxygen radicals. And these uncontrolled can damage DNA, protein, and lipids, which further potentiates existing lung injury or could cause new lung injury. But the lung protects against these effects by maintaining a careful balance of enzymes called antioxidants. And the antioxidant we began to look at first was superoxide dismutase, which protects against ROS by interacting with free radicals to produce hydrogen peroxide, which is then converted to water by either glutathione peroxidase or catalase. And what we found in vitro was that nicotine and cigarette smoke exposure alone induced significantly more SOD1 mRNA expression in pulmonary fibroblasts. Additionally, looking at the mice from the experiment earlier, we found that nicotine and bleomycin did induce SOD1 expression, but both of them combined further increased the amount of SOD induction in these mice, suggesting this may be one of the mechanisms by which mice are being protected against death. Additionally, this protection was completely absent in the alpha-7 knockout mice. Now, Similarly, expression isn't the only characteristic of superoxide dismutases. Activity is also important. So looking at either control mice or nicotine-treated mice, we found that SOD activity was significantly upregulated or increased in mice treated with nicotine. So that brings me to the summary of AIM-3, that we are showing nicotine is inducing SOD1, and this may be the mechanism by which nicotine is protecting against bleeding mice and induced mortality. So now we're going to get to the fourth aim, which is kind of shift gears. This is more clinical aim because I ended up meeting with Dr. Ralph Perez, who's not present here, but he had this idea of trying to improve tracking of pulmonary fibrosis in patients. And so we wanted to use the idea of, can we use this principle of increased matrix remodeling to possibly improve clinical tracking in these patients? And that's because right now, as far as non-intrusive ways to measure pulmonary fibrosis in patients, it's very difficult. The most common method used is called spirometry, which everybody has gone to the doctor at some point and took a big deep breath and then you breathe as hard as you can into a tube. That's called spirometry. And when you do that, it allows detection of several characteristics of your lung. And the two we looked at is FVC, shown here on the right, which stands for forced vital capacity, which is a measure of total lung volume, and which is currently used in the clinic. And the other value we began to look or try to include was called FEV1, which stands for forced expiratory volume in 1 second. Now this is the volume of air that you breathe out in that first second, which is a measure of how fast or do you have an obstructive airway. And the principle for this is a change in 10% from baseline in FVC is considered progressive pulmonary fibrosis in the clinic. And this is often long-term, which can happen over a year, seen here. And this outcome is used in clinical trials and also in the clinic. And our reasoning is our formula could detect a 10% change in these patients earlier. You could do two things. You might be able to change patient regimen or change medications earlier, improving those patients' quality of life, but it could also shorten clinical trials, which would save money for the pharmaceutical companies, but also increase the time that new drugs can get to market. And so we developed this new formula, which we've not come up with a really good name yet, which involves using FEV1 over FVC predicted and FVC actual, which every person has a predicted value for these airway measurements based off age, height, weight, and race. And so this is a matter of using existing values that are out there, but just kind of rearranging them to what is currently used in the clinic. And because pulmonary fibrosis, idiopathic pulmonary fibrosis, only affects about 100,000 people in America, we didn't have enough patients at U of L to be able to look at in-house. So what we did was applied and got access to the Panther trial database, which is a previous pulmonary fibrosis trial conducted looking at prednisone, azathioprine, and N-acetylcysteine for treatment of pulmonary fibrosis, which yielded 236 eligible patients that were either treated with NAC alone, N-acetylcysteine, or combination therapy. Now, it's important, there was no difference seen between the two groups in this trial for the treatments. So what we did was we took all patients that had reached a decline of 10% in their forced vital capacity over the course of the study, which was 60 weeks, and compared when they reached the forced vital capacity decline of 10% to when they reached the decline in our formula. And what we found was indeed that our formula was detecting significant changes in these patients earlier than forced vital capacity alone. So in this graph, forced vital capacity alone is shown here in this dotted line, and our formula is shown here as a solid black line, with the number of patients that reached that 10% decline on the y-axis and the weeks of the study on the x. And what I just want to highlight is that at the first visit, we were catching almost 3 times the number of patients in pulmonary decline when compared to forced vital capacity alone. And this is significantly different. So we are catching the decline in these patients earlier, so we may be able to predict when these patients are progressing and allow for better care and shorter clinical trials. We also looked at the other important characteristic of could this formula be used to predict rehospitalization or death in these patients. Sadly, there was no difference between FVC or FEV1 in predicting rehospitalizations and death, and this is by an ROC curve, which is a way of showing how prognostically accurate a formula is. But with these low values, neither formula is a very good predictor of rehospitalization or death. So that just brings a conclusion of my 4 aims, where we show that nicotine induces a transitional matrix with increased collagen type 1 and fibronectin, and this is through the alpha-7 nicotinic receptor. Additionally, we show nicotine enhanced bleomycin-induced lung fibrosis, and that it somehow protects against bleomycin-induced lung injury, death, but it's possibly due to the induction of superoxide dismutase 1, which is through the alpha-7 nicotinic receptor. And lastly, this new formula that we've helped develop could be a new prognostic formula for earlier detection of IPF progression in these patients. So as Dr. Roman alluded at the beginning of my presentation, this work kind of highlighted my interest in trying to predict or help improve patient care by tracking outcomes, and whether we can use those to provide better care, but also improve the trial of clinical trials, or the design of clinical trials. And that's been part of my internship, which has been with Revon Systems and Apellis Pharmaceuticals, which is involved using, or trying to determine clinically relevant disease outcomes for each of these diseases that we've looked at, and the improved clinical trial design. And this has also involved the development of remote tracking of medical outcomes, where patients could input these values at home. And then we can use these additional values to develop what we call a smart symptom tracker, which is basically artificial intelligence to help predict when patients are going to have bad exacerbations or their disease is about to progress. Additionally, not all, as I showed on the second slide, not all pulmonary fibrosis is idiopathic. And part of that involved investigating some other pulmonary fibrosis diseases such as Hermansky-Pudlik syndrome, which is a clinical review I wrote with Dr. Roman. Also, fibrosis can affect other diseases or other conditions, and one of the other ones is cancer. And with some preliminary work done by Caleb Greenwell in our lab, he took mice and either treated them with PBS or bleomycin, and then after forming pulmonary fibrosis, injected them with lung cancer cells. And what we found was significantly more lung metastases in mice treated with bleomycin, suggesting that if you have pulmonary fibrosis, you might have an increased propensity for forming lung cancer metastasis. And looking at different pathways, Dr. Roman wrote a research review paper on targeting the mammalian target of rapamycin in lung cancer, and investigated another protein called CRKL, which is a potential protein important in EMT, which is a marker of cancer. Increased EMT causes increased lung cancer metastasis. So that brings the end of my research. I just wanted to kind of end this on a small sentimental note, and I want to dedicate this presentation to this woman on the left, my left, you're right. Her name's Ann Voker. She was an important woman early in my life. She was an educator for several years, but she died due to smoking-associated diseases. But she's not the only one. I'm sure everybody here has. In Kentucky, we have one of the highest concentrations of smokers in the country. Not only that, over 6 million people died in 2011 due to smoking diseases alone. So with that, there's a lot of people I need to thank, including the Starship Roman Lab, which Dr. Roman, who has been an amazing mentor. Our lab manager, Jeff Ritzenthaler, aka the Magician. He has truly magical hands when it comes to lab experiments. Dr. Edelson Torres Gonzalez, who is our animal guy, he has helped me with just about every animal experiment you saw here, and he was tremendous help. Caleb, who is not here today, but he's a graduate student in our lab who has, if nothing else, provided comedy along the way. No, he's been more than beneficial and helped me carry out several experiments. And indeed, Ryan McAllister, who is a newer member of our lab, who is doing interesting research in nicotine with influenza. Also, I'd like to thank Dr. Zelko and Dr. Watson, who are the adopted family members of our lab. They're always there to offer constructive criticism or give me advice whenever needed, and they've been truly helpful along the way. I also need to thank Dr. Perez, who I've been working with on the clinical project, and he has been very insightful and very helpful. I also need to thank my graduate committee, Dr. Artiel, Dr. Barve, Dr. Scott, and Dr. Susi, for their input and helping me along the way of this journey. And I also need to thank IPIBS and the Department of Pharmacology and Toxicology and the T32 grant that I was a part of. And lastly, I need to thank my family, because without their support, I would not be here. And I'm sure they're in the back reading the newspaper. So with that, I'll take questions.
Questions or comments?
I know I didn't do that good of a job.
There's gotta be something.
So smoking affect everything.
It does.
Yeah, smoking affect the oral microbiota.
It does. And
Microbiota affect the lung fibrosis.
no, that's some of the work. Yeah, so some of the work Dr. Scott does, and there is interaction between the two. I've not personally done much research on it, but one of the cool new areas of research is, I know 2 months ago, I think it was out of Harvard, somebody's developed basically a lung on a chip. So they can do multiple pulmonary cell types on a chip, and they show that exposing that chip to total lung smoke showed high characteristics with COPD and pulmonary fibrosis. So that as the models to study this, I would say go up in complexity, but the ability, we're going to be able to tease and look at more effects of whole lung smoke. Because as you said, it makes it very difficult to look at when you're looking at 7,000 compounds that can affect just about everything.
Dr. Hunt? Or I thought.
So you hypothesize, very, very nice, but you hypothesize that nicotine reduces ROS, and you show evidence that SOD was induced.
So yeah, I guess, and maybe I didn't say it clear. We originally hypothesized that there would be increased mortality in the nicotine-treated mice when exposed to bleomycin. And obviously we saw the opposite effect, that there was less mortality that was protecting somehow. And so it was more investigating what were the potential mechanisms by which nicotine might be inducing protection. And so I don't think nicotine's driving more ROS. That may be the case, but what we're showing is that it's increasing levels and activity of superoxide dismutase, which is an antioxidant. So it's actually increasing the number of antioxidants which can protect against the ROS induced by bleomycin. Does that answer your question?
So in the lung decellularization method, you put cells on that and see if it affects IL receptors?
Yeah, so we've done some initial research, but it's one of the future, I guess, directions or future research we do want to look at, because one of the things that would be cool is if we could show that if you took nicotine-treated mice, took those lungs, and then put immune cells on the 3D matrix, whether you saw that same robust response that we saw in vitro. So that is one of the things we're looking at. There have been other labs who have done that and seen, now not nicotine-treated, but whether bleomycin-treated lungs or some other diseases, inducible models in mice, and they are seeing characteristics transferred to those cells, whether it's increased migrations of the cells or looking at profibrotic markers such as alpha smooth muscle actin or collagen fibronectin, some of these profibrotic mediators or proteins involved in fibrosis.
When you're granted, maybe I'm misremembering, but the alpha 7 receptor knockout mice,
Yes.
they had increased fibrosis following bleomycin?
They had,
Yeah. After nicotine?
yes. Which, although as I stated, I focused on the alpha 7 nicotinic receptor, but there is a whole, I think there are 13 different types of nicotinic receptors. Additionally, nicotine could be affecting other cells or other cellular processes because it can interact with more than just the nicotinic receptor. And one of the more interesting things is nicotine may have a dual effect in that it drives increased progression of fibroblasts, increased protein deposition, but it may affect the immune system in a very different way. So having immune cells lacking the alpha 7 receptor may alter, I mean, as you saw, there was no induction with nicotine, but they lacking the alpha 7 receptor may alter those cells and how they interact at baseline. Because inflammatory response is required in the bleomycin model to form fibrosis.
So you don't have a contribution of the immune system versus the.
Correct. I mean, we just looked at levels, but that wasn't part of my research as far as trying to modulate the inflammatory response or different inflammatory mediators.
Other comments or questions? It was all clear. If not, then thank you very much. I can have the committee members stay.